Oh, there you
I think it's going actually. Yeah. Okay. So welcome everyone to this population dynamics seminar meeting. This is the first of 2026, and I will start sharing my screen and present the speaker of today. Okay. So probably you can see my screen at the moment. I will try to put it full screen. So today we have Julia Schwartzman from University of Southern California, and the title of her talk will be Ecological Dynamics of Bacterial Groups. Some word regarding the speaker. So she got a PhD in microbiology in USA, University of Wisconsin, and then she did two postdocs at Harvard and MIT then. And she became assistant professor of biology at the University of Southern California, USA, where it is actually sitting at the moment. So she's interested in several topics. Microbial collective behavior and multicellularity and symbiosis are her main focus. For example, she studies from phenotypic heterogeneity in clonal population of cell to cross-kingdom interaction between microbes and multicellular life. And at the moment, she's focusing on several topics that range from dynamics of marine bacteria, kelp microbe interaction, seasonal and spatial dynamics of collective forming bacteria. And she uses different methodology, um, ranging from microbial ecology, cell biology, and going to population genetics and biophysics. So with this, I would like to stop sharing my screen and give in a possibility to Julia to share her screen. And from this moment on, the stage is yours, Julia. Thank you.
Fantastic. Well, thanks for the very kind introduction, Marco. And please, if people came in a little later, interrupt me along the way. I'd much rather address questions as they come up. It's always more fun, especially when you're on Zoom, to have interaction. Okay. I gather that I'm the first for this year's series, which again, I was saying like it's a great lineup. I hope to come back. I mean, if it's okay, I'll come back and see some of these. Um, as Marco said, I am interested in bacterial collective behaviors. I'm really interested in spatial questions. I was laughing because I have broad interests, but really my lab is focused on, you can think of it as a microcosm, a coastal ecosystem at the micro scale, and we study questions that are bacteria interactions, and then bacteria and actually kelp interactions, which is no kelp in this talk, but I'd love to talk with any of you offline about that. Okay, so let's see if this is going to play. All right. Um, just to kick things off, I don't think I need to introduce anybody in this room to this idea, but kind of the starting place for a lot of the things that I think of in my group is the fact that bacteria engage in a diversity of collective behaviors. And some of these might be familiar to you. They range from density-dependent chemical signaling, like quorum sensing and signaling, phenomena like collective motility. Here you're seeing, I believe it's Proteus swimming out on a swarm plate. And then, kind of more natural examples on your, in the dental plaque biofilm, there's all sorts of very interesting self-organized spatial patterning that emerges as the members of the plaque microbial community self-organize. Okay, so all of these are examples of collective behaviors, but only some of them are accessible to study in the wild. You can scrape plaque off your teeth, but it's not a very dynamic system to study. So this creates a challenge that my group is very interested in addressing, which is how do we link the form of a collective behavior to its ecological function? So we often study, there was a great debate, if you ever followed this in the quorum signaling literature, about whether it was density dependent sensing or something else. And that in part arose because it's so hard to figure out where the context is that microorganisms are engaging in this signaling behavior. So really the challenge, the context challenge, this is kind of a genotype by environment type challenge. There's cellular phenotype and behavior that's possible for any given microorganism, but the behavior that's actually expressed in the environment has to do with, it's the cell interacting to its environment, and sometimes, depending on the context, shaping the environment itself. And so there's a feedback, which I find very interesting. Okay, so the focal example of collective behavior that I'd like to spend some time on today with you all is this idea of cooperation, so density-dependent growth. This is a really, really, really generic ecological phenomena. In bacteria, in the model that I'm going to introduce you to, we can think of it as, this is a very spatial thing. A lot of bacteria that eat organic matter. It's not like you would study in the lab. Maybe it's not glucose or some other simple resource. It's a complex resource that's often, bigger than the cells themselves. And so if the substrate that is resource limiting needs to be accessed by a cell that's too small to transport the substrate, cells often secrete extracellular enzymes, right? And so you can think of density-dependent cooperation in this context as the ability to pool enzymes and share resources. And so in an environment where the resource is hydrolyzed outside of the cell and some of it might be lost to diffusion or not accumulate otherwise, it's possible for an individual cell, let's see if I can find my cursor, it's possible for an individual cell to not be able to produce enough enzyme to recover sufficient resource to survive. And so this creates a really interesting density-dependent threshold, growth threshold. If cells start to cluster, they can share resources and more efficiently create share enzymes and more efficiently grow. And that really leads to this increase in per cell benefit over the size of a group. Of course, there's some upper limit to group size, the benefits of group size. At some point, too many cells in proximity to each other are going to start to compete, right? Okay, so this is positive density dependence. You might have heard of it referred to using that term, or perhaps maybe the Ali effect. That's more in the ecological literature. Okay, but this is a really, really, really well-characterized phenomena that happens across, all parts of the tree of life. All right. In marine systems, it turns out that organic matter, so these complex resources, there's a lot of marine organic matter out there. You might have heard of marine snow or marine particles, but the truth is, so I'm in a department with a lot of biological oceanographers, and if you talk to them, they'll say, well, organic matter, it's really chemically complex, but also the size of organic matter is very complex. It's not just big particles and really small resources. It's a continuum, and it's actually something that chemical oceanographers study. They study how it condenses, and you can think of it as this really complex resource or substrate that ranges a lot in size. And so a lot of marine organic matter is it's in that kind of odd size class where it's not exactly a particle, it's not exactly a cell sits down on a substrate, but it's much too big for a cell to directly transport. So you can kind of think of this as a lot of resources are on the same size scale as the cells that are foraging them. I'm going to pause there. Do people kind of nobody's completely lost yet? Okay, it's interesting because I think we're so ingrained in thinking about really small resources or really big resources. I guess, what I want you to take forward from this talk is I'm thinking about kind of medium-sized food or resources.
So it's similar to like cellulose degradation, right? In some sense.
Yes, yeah. And cellulose, cellulose can be a really big particle.
Yeah, it can be small. Exactly.
Exactly. Yes. Yeah. Hi, Wenying. Nice to see you.
Yeah, sorry.
Oh, amazing. I saw some familiar names. Thank you all for coming out for this. All right. So yes, medium-sized food, to put kind of casually. All right. So we're in this regime. So this is a system that I started to develop when I was a postdoc with Otto Cordero at MIT. Otto, as many of you know, has this wonderful collection of marine isolates that he built in part from working with Martin Poltz. And so we took some of them. I have a kind of a dark history studying Vibrio. I somehow always come back to this as my model organism. It's got a lot of many wonderful features. And so Vibrio is a genus of bacteria that degrades. They're very good at degrading polysaccharides. And there's a lot of variation kind of in the trait space of how that's done. And so we're using one of the polysaccharides that Vibrio degrades frequently is alginate, which is an abundant marine polysaccharide. It's produced by kelp, actually, strangely. But you can make, if you've ever had boba tea, it's a really good hydrogel former. That's one of the things that boba can be made out of. And so if we put alginate into, I'm just showing you here 2 different strains of Vibrio, and I'm putting some alginate into the plate, and you can see in the background, if you can see my cursor, it's slightly hazy. And then I've spotted colonies of these 2 different strains onto the plate with alginate. So this is a halo assay showing that if cells in the colony can secrete alginate degrading enzymes, then they can break it down. And so you can see that there is variation in this trait space with regards to the kind of time scale and the strength with which alginate is broken down that varies across strains. And so we know from work from Martin Polz's lab and Jan Hendrik Hegemann that there's really been a lot of like the adaptive radiation of Vibrios is often shaped by how they degrade polysaccharides, and in particular, some species, it's really been shaped by how they degrade alginate. Okay, so very variable trait is what I want you to take away from this. We also know a lot about kind of the biochemistry of how alginate is transported into the cell and broken down, which is another good reason that this is a useful model system for us. Okay, so moving from a plate into kind of liquid culture and closer to a marine system, if we grow these 2 different strains that I was just telling you about, this is old work, so I'll skip over it. But when I was a postdoc, we started to do this. We wanted to think about how different strains break down alginate and the extent to which they do so cooperatively. And so if you compare these 2 strains and you start with different initial population densities, you can see that strains that on a plate, or a strain that on the plate weakly degrades alginate, it can grow, like larger initial populations can grow, but at some point the growth rate slows down. And then there's a initial population density below which you don't get growth at all. And so we think that this is evidence that some strains can grow cooperatively on alginate, whereas, as you can see, others don't have this density dependence. Right at this threshold where this particular strain is starting to struggle to grow, we noticed a really interesting phenotype, which is the cells start to form groups and they start to clump. Okay, I'm gonna minimize something. All right. And so when the cells are starting to form aggregates, one of the things that we noticed is that depending on the initial density, they can do so either clonally or aggregatively. So cells can start from one initial individual and divide. And you can see if you quantify this, you can quantify clonal growth if you take aggregates and you genetically label them. So these are isogenic strains. They're just labeled with different fluorophores.
Are they motile? Are they motile?
So sometimes they're motile. In this case, that's a great question, Wenying. So yes, in principle, they can be. In this system, it's a shaken flask, and I don't know the time scale over which they're retaining motility. Does that answer your question?
Yeah.
Awesome. Love it. Okay.
Hi,
So
Julia.
in this case, yes.
Can I interrupt you? Can you remind me, maybe I missed it, how you give the alginate? Is it just molecules or is it aggregates? Because then if it's aggregates and the shape of the aggregates, how porosity is going to affect the threshold growth,
Yes.
right?
Yes. So Marco, thanks for your question. And hi, by the way.
Hi.
So you could, in principle, what I like about alginate is that you could make a landscape of different size things. What we are doing in this case is we're taking so alginate sold by viscosity or DP, so degree of polymerization. And so we're taking alginate polymer that is sold as medium viscosity, so it's in a range of kind of smaller sizes. And we are dissolving we're dissolving it as much as possible. And so it's kind of, I don't mean to give you some sleight of hand, but it's a long polysaccharide, but like not long enough that it's gonna condense into something huge that the cell can sit on.
But it's going to make a blobbish, right? So it's going to have a radius of gyration, which is more or less what size?
So it should be about 1/10 of the size of the cell. So you can kind of go from, it's about 1,000 Daltons, and then if you think about the size that might be, then it's they're small blobs is my best guess. And we're also, I should say, thanks for the opportunity to clarify, we're dissolving just a teeny little bit of alginate. It's definitely the resource. We're carbon limited, so we're limited by alginate in the system. But we're not, if we dissolve a lot of alginate, then you start to get into this regime where you think about how the alginate's condensing, and we'll get into this in a minute, but the encounters start to be a thing. But we think it's kind of an isolated, patchy system of kind of these blobs on the timescales that we're worried about. Does that answer your question?
Yeah. Thanks.
Yeah. We are very interested. I think you could use like maybe DLS, like dynamic light scattering, to characterize the size distribution of the alginate, and we haven't gotten there yet. Yeah. Okay. Cheers. So there's a regime in which we can grow these strains that show strong positive density dependence. If we start with a small enough initial population, they'll grow clonally. If we start to increase the initial population of the cells, they start to do really interesting things. I told you that if you start with a large enough initial population, they can form aggregative structures. So instead of one cell dividing into a group, cells find each other and then they can grow. And what I'm showing you here is my postdoc Tom has actually started to look at, you can form these groups of groups. And so we think that in our system, it's really, there's stickiness, there's a tendency to aggregate, but there's also, as you can see, this tendency to encounter. Okay. So moving forward. So thinking about, I hope I've given you some intuition, and thank you for the clarifying questions, about kind of the properties of our experimental system. So now I'm going to try to do something fairly ambitious.
Sorry a second, Julia. There is Thomas that raised their hand. Can you speak, Thomas, or would you like to write in the chat? Thomas?
Can you hear me?
Yes, we can. Go.
Okay, great. Thanks. Thank you. I somehow missed this point. When do they aggregate? When do they clonally? At high densities, low densities?
Whoops, sorry. So when they're clonal, if you start with they can grow clonally if you start with a very small initial population density, and they'll grow aggregatively if you start with a larger initial population density. And we'll get into this in a minute, but the intuition is basically encounters are concentration dependent. And so you can get them, they can form groups, they can find each other if there's more of them initially. And
okay.
But they also have this tendency, as you can kind of see in the inset that I'm showing, this strain has this tendency to grow in a sticky way. And so they can also grow and stay together to some extent. Yeah. Fantastic. This is all context dependent. Sometimes when I present this to people, they think, oh, the strain's always sticky. It's going to be, no, if you change the context, these things, if you grow them on glucose, they're single cells that when they swim around. So this is one of the features and bugs of our system is context. Okay. So like I said, I'm going to do something fairly ambitious and try to relate this dynamic in this system to what might be happening out in nature. This was really inspired by when I started my group. My colleagues go out into the field a lot, and they had this great conversation with Jed Furman, who some of you might know. And he said, this is a really interesting phenotype, but like, how do you know that, this is to paraphrase Jed, he doesn't talk like this, but how do you know that this is happening in nature? Which is a very valid point. Okay, so we're going to try. I'm not telling you that I've solved this entirely, but this is kind of where we're trying to head. All right, so thinking about the microscale, and really the environment that I'm trying to mimic is like a coastal ocean. I think I've already introduced you to this idea, but I hope I can convince you that the way to think about a system like this is that it's a patchy resource landscape. And so there's nutrient hotspots. We're not kind of in a well-mixed regime. There's nutrient patches that cells can navigate between. Roman Stocker's group has done some really elegant work, and Thomas Kirbo and a lot of people in the marine space have really been thinking about these systems as kind of patchy landscapes. Okay, so from the microscale, I love to show this slide. Like, people have been thinking about what happens at small scales for a long time. There were some experiments done 100 years ago in freshwater and marine systems where you sunk glass slides into water and then, sometime later brought them back up and looked at cells that had encountered the glass slide. And so you can see that there's actually a lot of stickiness in a system like this. There are a lot of cells which are shown in these are the original micrographs, so the dark things are cells. And they're cells that can actually adhere to the glass. And this is what gave us the first concept of biofilm. But what I want you to take away from this is, yes, biofilms can form on a surface, but it's the system really is cells swimming around and encountering. And a lot of cells that encounter a surface, they can stay and they can form a biofilm, but it's just as often that they'll stay for some time and then leave before actually committing to a surface and making extracellular matrix and everything else that you might think. And so like a lot of the cells that you see here, there are not a lot of them, and you don't see evidence for a lot of matrix. Okay. So encounters, that's what I want you to take away. There's a really nice framework that my colleague Jonas Slomka, who actually just got his own independent position in Poland. He was a postdoc in Roman Stocker's lab. So Jonas and other people in Roman's lab have been thinking about this idea of basically how are cells shaped by the fluid environment. And they're using this framework of encounters. And we'll get to this in just a second. So I was really inspired by their work. They were thinking mostly about kind of individual cells and how they navigate a fluid environment. And so I thought, well, in our case, maybe this is a useful framework to think about how cells and groups of cells encounter an environment. And so Jonas's idea is to use this concept of a physical encounter kernel, which you can think of. Their hypothesis is that you can decompose this kernel to reflect different aspects of the fluid environment. So there's a kernel that could describe diffusion and buoyancy, turbulence, and kind of under the hood of all of those kernels, the units are volume per time. So it's how much volume is sampled over time by an individual thing. There's aspects, there's a diffusion coefficient, there's size that are, going to shape what the actual nature of each kernel is. But if you sum them all together, and this is a hypothesis, you can kind of reflect what the net encounter might be in a given kind of volume of fluid. Now, in addition to kind of the composition of the kernel itself, these also depend. You can imagine if you have two things sampling a volume, you can think about just those two things, but you can also think about a system of two things, and then it's going to depend on the concentration of both populations. I can build up on this, but please ask me questions if this is confusing. The kernel aspect, I think, is sometimes a little complex. Yes. Yeah.
So I'm not entirely convinced how they can all be additive. Like, I mean, how are there no effects that counteract each other, for example?
Oh yeah, so definitely this is, I would say, probably a coarse grain estimate. The intuition that I want to give you, what I'm going to present moving forward, we could just think about this as a diffusive process. We know we have a shaken flask, and so we know that that's not quite the case. But I agree with you. This is a hypothesis, and there are very reasonable, concerns about, especially when you think about, there can be interactions between the processes and kind of this length scale and everything else. So yes, basically I agree with you. Yes, but I agree with you, but what I'm going to show you is kind of robust to this, and we could use we're not going to rely on our estimation of the exact encounter kernel for this. All right, so if you look up conditions in the ocean and use a kernel that's drawn from there, then you can make a phase space where here we're going to think of one microbe encountering a population of, I think it was 100 different resources, just so we could have some encounters. In a volume, in a given unit time, we're asking, how many encounters are happening. And so the magnitude of the kernel is what we're plotting on this phase space. And so what I'm showing you here, just want to build some intuition. there's different regimes that emerge. And so if you think about kind of the first regime, or this is, I guess, the first regime, we're thinking about particles or resources that are larger than cells. And so this is a regime that's kind of more, sorry, I need to advance my slide, more like marine particles, so a particulate environment. Here we see that the magnitude of the encounter kernel, and therefore the number of encounters, is going to increase as the resource particle gets bigger, but the size of the microbe is going to be less important for the number of encounters that are observed. And so basically in this regime, it doesn't really matter what the size of the cell is. If the resource is larger than the cell, they're going to be more, the size of the resource is going to drive the encounters. We can think of a second regime, which is really well characterized, that is more driven by the diffusive part of the encounter kernel. So they're small resources and small cells. These are kind of ephemeral. This regime has kind of been described in these ephemeral environments. Roman Stocker's group has done a lot of work in this area. There's a third regime, which I would argue hasn't been really studied as much. That's not particulate, not ephemeral. It's this intermittent size regime. And so I guess I would define this as the resources are smaller than cells, but not so small that they're kind of just diffusing around. In this regime, you can see that what really matters is the size of the microbe. That's going to have the dominant effect on the encounter kernel. And so as the microbe gets bigger, and you can see we're pushing this into kind of, it's a radius. And so you're not going to get a microbe that's 1,000 microns wide, but you can get a microbial group. And so we think that this regime, this type of resource, is maybe one where forming a group would provide potentially an advantage for populations. Okay. So we wanted to, I mean, this was great. We dove into the theory that Jonas and others have been working on with encounter kernels to kind of give us some intuition for what we might expect. But I'm an experimentalist. And so really what we thought we could contribute was to use our system where we do have bacterial groups of different sizes to try to test this intuition that, groups and encountering resources that are, not tiny, but not huge, that there would be some increase in encounters with increasing group size. And so what I'm showing you here is an experiment that my postdoc Tom did, where he's taking one of these group forming Vibrio isolates and he's growing it in the presence of alginate, like Marco and I were talking about. It's small alginate, and because the alginate itself is like long strands and it's a little hard to characterize its exact size, we've spiked in these model resource particles that are 1 micron in size and they're spherical. And we can change the surface chemistry. I'm happy to tell you about the controls we did around that. But really the result is that larger groups over kind of an encounter kernel type time in a set volume, larger groups encounter more resource particles, which is great. That and we can come up with, we can fit this kind of distribution of cluster sizes and resource particles and come up with an empirical encounter kernel, which, for the purposes of where we want to go, I think it is interesting to try to relate this to kind of what we're estimating and what we were modeling before, but I think it's going to be very hard to kind of directly compare the two. So basically,
just a quick question. So cluster radius is a cell cluster.
Yes.
Cell cluster. And the resource particles, it's like if you, so what does the Y? So basically, if you have bigger cluster, you have more resource particles.
Yes,
I see.
A bigger cluster moving around in the volume.
Oh, and then you count number of resource particles.
Yes, because they're fluorescent.
Oh, they're fluorescent. I see.
Yes,
Okay,
yeah.
Great.
It's tiny on the screen, but right. So we have clusters and we can put in fluorescent resource particles. You could play lots of different games if you wanted with different colors and different sizes or whatever.
So the green stuff, is the green stuff a resource, the green dots?
Yes, those are, and And for the purposes of being able to wrap my head around the quantification, we're using these spherical polystyrene beads.
Oh, I see.
Just so that it's, there's not kind of an uncontrolled variable of there's a size distribution and they're colloids, they're not anyway. So basically we're artificially measuring encounters.
I see.
Okay, fantastic.
Julia?
Thanks. Yes.
I have a couple of questions. Can I interrupt you here?
Please. Yeah.
So the first one is in just normal diffusion, right? If you have an absorbing sphere, the flux onto the sphere depends on the radius, right? So it grows linearly with the radius. So do I understand correctly here that your scaling would be instead to this actually more than the surface? Is that the power 2.6 here means that is the scaling is higher than radius squared? Is radius to the 2.6? Or
yes.
Maybe I didn't understand well what the source particle is.
Yes, yeah, that is the expectation. And I think that, this is the empirical kernel that we're measuring. You would expect it to be to the power 2, I believe, if it's just diffusion. And if it's more than diffusion, it should range between like 2 and 3.
If it's just diffusion and there is no flow, it should be power 1.
Power 1, yes. Sorry, this is.
But
Marco,
I think Marcos, aren't you talking about the flux to a sphere? But this is like how many particles the thing meets. It's not quite the same thing.
No, but they stay attached, right? So it is equivalent to a perfect absorber, as far as I understand.
No, but yes. I think it was right. Like it should be this to the power of 2. So
it should be to the
cluster
power
surface.
Of 1. It should be to the power of 1. With diffusion, is power 1 for sure. It's a textbook example that you do in undergrad courses. I'm talking about diffusion equation with a perfect spherical absorber is power 1, is not power 2. But this is power 3 almost.
Yes, but this is, I think, so I will say that this is, being an experimentalist, you are probably more versed. I'm not a fluid dynamics person in truth. But the expectation, this is like a spherical, we're in 3 dimensions. And so, I think this is kind of in the range of what we would expect for, the other, like a kernel. It should range between 2 and 3. That's not a very satisfying answer, Marco. I'm sorry.
But the other thing, which is more biological, I wanted to ask was, if you imagine these little green dots as being the nutrients, right? This is what you were thinking. How is the thing that's shared through the population that makes up the cluster?
Yeah. This is, yes, that question. Okay, that question I have lots of thoughts on. So we're assuming here at the moment that, and we're going to get into this in a minute, that you have encounters and then magically the resources partition across all the cells. That is obviously not the case. We know that that's not the case in our system. And so you can imagine, we'll get into this in just a minute. But this is, on the face of it, one of the, well, actually, can I advance a couple slides and then I think I won't repeat what I'm just about to say. Sorry. Okay, amazing, thanks. But yeah, I should have a better answer for, sorry. This is about to give you better intuition about the scaling and why this is the power that it is. I guess unsatisfyingly, I will say that we think it should range between about 2 and 3. Okay. So, that was kind of total encounters per population. If you think about encounters per capita, and this is getting directly to Marco's question, what we see is that, so if you make an estimation of, you have your group, and we kind of know the packing density of the cells in the group, and so we can estimate how many resource particles per cell in a given group. We can quantify, we can make an estimate of this from our quantification. So you can think about how many resources are available to each cell in the group. And what you see is, even though more resources are encountered by larger groups, if you look at kind of the per capita benefit, you see that because of the fact that it's a volume and because of the scaling, as larger groups per capita, there starts to be a disadvantage for cells in groups. As the group grows in size, the resource available per cell decreases. And so, what this means is that there's not, I mean, it's a little surprising because it's more resource, but per cell, they're not doing as well as they might if it was an individual cell encountering a resource particle. That said, I want you to notice that there's a lot more variation in resource encounters for smaller cells, and that kind of makes sense because you have a population of individual cells, and some of them are going to encounter resource in kind of the unit time that we're thinking of, and some of them are not, and so it's much more stochastic. And so it's kind of individual cells can hit the jackpot, or they won't, and there's more variability, versus large cells in larger groups are always going to encounter some resource, but it's going to be suboptimal relative to what they might encounter. Okay. So this is just another view of this. And so we're thinking that this is really an interesting kind of trade-off between consistency and amount of resource that could be encountered. And so we can quantify basically the consistency as the inverse of the amount of the per capita amount of resource. And I think it's a really interesting trade-off, and we're just beginning to scratch the surface. But if we kind of plot consistency versus amount, and we kind of look at the shape of the distribution, we think this is putting my neck out a bit, but we think that it's a convex concave relationship, which would suggest that maybe, and we would love to test this, that it's, if you were to evolve the system, for example, that the kind of the optimal solutions lie on either end. It's you either want to be really big as a group, or be lots of individual cells, but kind of the middle state of intermediate size groups.
Julia, I'm curious, right? Whether you have measured like the like a total population growth rate, right? Like with small cluster versus large cluster, right? Like sort of like you sort of like sort of like averaging. Because you were talking about I mean, which one is a better strategy, right? Like a shaken flask, either it's consistent with small ones, like small clusters or large clusters, and like looking at the degradation rate of alginate or the growth rate of the total biomass.
Yeah, we can definitely quantify the growth rate of the populations, certainly.
Yeah. And which one bigger? Yeah, which one do you expect to be bigger?
I mean, I guess I would expect I think, okay, there's also something about speed. I think that the cells that can grow as individuals are probably going to do so quite quickly. But, so I think that, how do I say this?
Or at the low nutrient. So like, I guess it's also like to do with it, like with the nutrient level, right? So it's a cluster size and the nutrient level, right? So it probably should be a function of these two, right?
I agree. Yeah. And so we're just about to do this. So we have this large collection of natural isolates, and this trait does vary quite a bit among the isolates. And so I think that that's the entry point into this, is to kind of common garden in one environment, one resource limited environment. What you're going to get is kind of a range of cells that grow as small groups and kind of stay small, cells that grow as individual cells that are, or the group, things that form really large groups. And then I think quantifying, it's not entirely clean because it's not the same genetic background exactly, and so there's some other aspects of growth, but I think that that's kind of but I agree with you, that's a really interesting question. I think evolving the system too, I would be very curious to see if it evolves,
especially if you can label different strains with different color, then you can look at whether this switch strategy could be really cool. That's right.
Would you review the grant that I just submitted? But yeah, I agree with you. This is, so thank you. I agree that these are all kind of interesting questions that you can ask, but I think the utility of, thinking about, I think that there's a lot to dig into with the encounters, or the intuition that you get by thinking about this physical encounter process and whether it's stochastic or consistent, you know, and how that's size dependent, I think gives you a lot of really interesting threads to pull. Cool. Okay.
Sorry, we have
Could
a
I ask
couple
you a
of
question?
questions.
Oh.
Yes.
Can I go
Yeah,
first?
please go. Please go, yes.
I was wondering, is this behavior completely genetically determined, or can they sort of decide to stay on a cluster and maybe then at some point detach and go jump ship, so to speak?
Okay. So I didn't quite hear the first words, but just to make sure that I'm answering the right thing, are they kind of do they stay stuck, or is this a variable trait?
Yeah, so you mentioned that you had these different strains that have different tendencies to cluster or not. I was wondering to what extent is it really like a static thing, or can they decide based on conditions, cluster size, whatever?
Yeah, they can, right. So there's, they're not, they can decide or not decide, but like they can, it's a variable thing. So you can, the clusters, let's see if I can find a good example of this quickly. Just going to skip ahead and show you. So these clusters are usually formed of a mix of cells that can either be motile, although we're not sure if it's flagellar motility exactly, or they can be stuck together. And so, and the proportion, like over time, the proportion of kind of cells that are stuck together versus cells that are swimming can change. And so to form a tightly packed group, it's, you know, most of the time tightly packed, but strains vary in there. Sometimes like these groups, like the green blob that I'm showing you, if you were to look at that over time, it's shooting off individual cells, or here, sometimes you can get quite a substantial population. I'm showing you kind of mixing emerging in the middle of a group, and then you get larger kind of dispersal events, kind of this kind of like a typical biofilm cartoonish view. The whole population on the inside gets released. And so really, you know, this is a very long way of saying it's a very plastic trait. And so you can have, you know, and it's very context dependent. And so in the environment that we think that the Q is actually resource limitation and starvation. So in the particular environment where the resources are limited, this particular green strain is going to be very, very tightly packed. But as the environment changes, that constraint can relax. Does that answer your question?
Yes, thank you.
Yeah. And so I think that it's a very interesting, and I mean, I think it makes sense because these strains are navigating changing environments. And so to me, it kind of, the Just So story is like, okay, yeah, I'm not surprised. There's a lot of regulation that happens. Okay.
Sorry, we have two more questions from the chat, so we'll read it
Yes,
aloud.
love this. Yeah.
From Justin Gom, we have how do resource availability and environmental condition induce fitness cost in bacterial population growth or clustering?
Is there fitness cost? So I think, I mean, we haven't directly, one way to ask this is to compete strains against each other. So I think that our prediction would be, yeah, there would be a fitness cost that has to do, if like the group, there's a fitness cost of being in a group per capita, but that might be balanced by, it's an interesting population level question, because if you're just thinking on the individual cell level, there's a clear fitness cost to being in a group. But at the population level, there might be, you know, it might be more, it might make cells more fit for all of them to get some resource, even if it's not as much resource as they could get otherwise. And so there's some really interesting population dynamics questions there. I hope that answers your question.
Thank you. There is another one from Tanis Patodi. Have you checked the distribution of the cluster based on the number of cells? Maybe there might be an optimal number of cells that can be in a cluster for optimal growth.
Yeah. Okay. This is great. This is going to get me into my next question. So we have measured this. Actually, Tom came from a group that was very interested in cell packing. And so that was like the number one thing on his list of traits that he wanted to look at. There is variation in cell packing among strains that we look at. But in terms of optimality, you know, I think it's really going to depend on kind of the rate of resource consumption and other, there might not be one true best packing. So you can imagine, getting back to kind of this trade-off between consistency and amount, you can imagine that you can overcome the kind of amount aspect of the trade-off if only certain populations of cells are allocated resource. And so if you have a whole spherical volume and only a fraction of the population is actually consuming resource, then that fraction, that population fraction is then going to get more resource per capita than they really should if you're just doing a simple allocation. And so that can affect, that's not packing, but that can affect kind of, these allocation questions. I could imagine that a less dense packing, like another way to overcome this trade-off is, instead of having everything tightly packed, you have kind of a poofy, like the configuration. I sometimes say honeycomb, but that's not quite the right way of putting this. But if the cells are less densely packed, then the allocation per capita can be increased, and you can overcome, they can be large, but like less dense. And so, right, I think to the question, I think that there's some optimality, but I think it's going to depend on kind of like the metabolic rate and like how many are dispersing and leaving. And so I think it's a little complex. Hopefully that answers your question. Please follow up. I didn't see who asked it, but okay.
Wait, there is another one actually with a follow-up.
Okay.
Are there only one type of strain in the system in this case? So he's asking Tanis.
So right now in our experimental system, we are looking at the growth of single strains. I think it is quite interesting to think about kind of more ecological populations. So groups of strains competing and potentially interacting, and kind of, and that's a direction that we're moving towards. And we have, as Wenying alluded to, we have the ability to do simple genetic modifications, so we can label strains, we can combine them. But right now, this is the complexity of, with all the phenotypic variation and everything, the complexity of one strain is enough for us at the moment. Okay.
Yeah, thank you.
Of course, yeah, thanks. Okay, so we have this trade-off. We think it would affect, some strains might grow as single cells, some as groups. This is just one strain, and it's kind of like allocation through time. All right, so if we're thinking of ways to, things that we know are kind of, biology fights a trade-off is maybe what I would say. And so we know that this allocation of resources isn't even in when groups are tightly packed. I have some previous work showing that there's a lot of phenotypic variation that emerges as groups that grow as tightly packed spheres get larger and larger and larger. I did some nanosims with Victoria Orphan's group at Caltech, showing that there's gradients of kind of allocation. This is incorporation of ammonia into cellular biomass. And so there's some really strong gradients that emerge kind of from the periphery of the group to the middle of the group. There's, more incorporation of heavy labeled ammonia than there is in the middle. And then this is, correlation does not necessarily equal causation, but also we observe that there's kind of phenotypic differences between kind of the populations that are in the center versus the periphery. And it's not unreasonable to suspect that there's some kind of nutrient limitation based cues that could be triggering that. In particular, we see different allocations of carbon and into storage granules in kind of subpopulations of cells. So these are very phenotypically heterogeneous groups. And we think that in the context of this consistency amount trade-off, that, we think that in a just so story way kind of makes sense, because groups aren't getting the allocation that they really should get. They should be a little more resource limited than individual cells. Okay, so I want to, sorry, this is great. The questions are great, and it's totally fine if we don't get through stuff, but I want to, just briefly touch on this. Somebody asked if we were thinking about multiple strains in the system experimentally. No, we have started to think about this using some simulations. And so you can model this kind of trade-off using a really simple model. So we'd like to really start simple and then add in complexity. And so imagine that we have a simulation where you have two different types of cell that can be competing for resources. The different types are going to basically grow exactly the same way based on Monod kinetics. And they're either going to grow, and when they accumulate enough resources, they're going to divide and become two things, or they're just going to grow and become bigger. And the size and the number are going to relate to how each population is going to acquire resources. Okay. So in a system like this, we wanted to ask, is there ever a regime, because the larger groups, the cells in the larger groups aren't getting as much resources, our naive assumption was that, well, the whole population of cells in those groups, they're going to grow slower. Maybe there's no apparent advantage to growing in groups. And so are there regimes where an advantage emerges? Okay. And so we can see that if we there's two different ways that you can think about the simulation. For each of the populations, you can say that, I'm going to say that all of the cells or groups in that population are encountering resources exactly the same. They all get the same allocation of resource per time step. If that's the case, then we think of it as an environment where there's no fluctuation, there's no stochasticity, then there are almost no regimes where a multicellular group, a larger group, is ever going to be advantaged over a population of single cells. If you instead allow the simulation to run and allow resources to kind of, more stochastically encounter, so that the population of individual cells, some of them never encounter resource, but some of them encounter a bunch, whereas in this case, the groups are going to more consistently encounter resource, then you see regimes of kind of physical encounter where we predict that a population that can grow in a group will have an advantage. And of course, this is just a prediction, and there's a lot of things that are, maybe not entirely reflecting the intricacies of like what would happen in an experimental system, but it's a prediction that we can start with, and that we're thinking of carrying forward in our experiments. Okay, we can modify, we're starting to play with this a little bit. You can imagine in a model like this, you can add in other traits that the single cells could have or that the groups could have. So for instance, if you allow the single cells to swim, you're effectively allowing them to encounter more area per time, and therefore encounter more resources. And so if you think of just a really simple swimming as kind of having a higher rate of diffusion, you can overcome this in a stochastic environment. If the individual cells swim, then you can start to balance this apparent advantage of being a multicellular group. You could also imagine if you give the group a growth penalty, then the population of individual cells might gain a larger advantage. Or somebody asked this question before, which is great, if you start to play around with cell packing, then you can advantage the groups slightly more. So it needs to be backed up by experiment, but we're starting to pull the thread of maybe how do kind of variations in traits among strains shape this apparent trade-off, and can we start to predict which traits are going to be more advantageous in a given patchy resource environment. Okay, so I kind of brought this up before, but there's a lot of variation in traits. There's a lot of phenotypic variation within groups, but also there's genetic variation across strains, which I think will be really interesting to play with as a system. We are sampling. So there's a bunch of Vibrio isolates out there from Martin Pultz in particular, and we've used some of those over the years. But my group, since starting, has built up a substantial collection of strains that are sampled from, excuse me, out in the middle of the ocean, well, off the coast of Los Angeles, kind of 900 meters down and at the surface. And we think that, there's some really interesting seasonal population structure in these groups. And it's they're ecological populations that can form groups when they grow on alginate. We think this is going to be a really interesting experimental system to carry forward. Okay. So thinking out in nature, I know we're running up on time, but I just want to end with this thought. Of course, this trade-off between this encounter-based framework, you don't have to think of it as just cells and groups of cells encountering resources. There are all sorts of things to encounter that are not resources, like bacteriophage, other toxic particles, other cells. You could, make the system more complex. We have taken a step down the road of, you would think it would be just inevitably bad in a marine system where phage are thought to outnumber bacteria by 10 to 1. You would think that this would be an immediately bad thing to be a group. And it turns out that inevitably, yes, groups will be infected by phage, and we can play with the infection dynamics and the burst size. I'm happy to talk to anybody about that if you want to know more about kind of what we're thinking. But the one thing I want to leave you with is this idea that yes, there will be an infection, but what matters is how long until the first infection. And a population that's in a group versus a population that's distributed as individuals, you can have different kind of dynamics of how the infection sweeps through a population. And so things that are prediction, I'm just telling you the digested kind of what we're thinking, not how we got there, I apologize. But things that are in a group, it can take longer for phage to find them, and the infection dynamics can be dampened. And so there are kind of advantages or differences in infection dynamics that emerge that we think could be quite ecologically relevant. And so the question is really, how much food can a group encounter before it encounters a phage, not how do groups necessarily evade phage altogether. Okay. Sorry, I want to make sure that I respect everybody's time. This was already covered, and I should have shown this slide but just for Jerome, I think you asked, are there a mix of single cells and groups? There is a lot of heterogeneity. There's a release of cells from groups. And so we think this kind of encounters with phage versus encounters with food, we think in this case, we think group formation kind of has to be quite dynamic, and maybe it might involve kind of release of a group forms and releases individual cells that can go on to form more groups rather than stay kind of as an obligate group over, long periods of time. So we think that this is kind of heading in a kind of cycles of group formation direction. Okay. Groups, let's see. And so just to end, I apologize, a little over time, but the questions are awesome. So I want to put this into a larger context. What I've told you about is how individual cells and groups encounter resources that are kind of medium size, about the size of an individual cell, like micron size. But there's a whole other landscape of resources that can be encountered in environments that we can think of as patchy. There's been a lot of great work specifically in Vibrio about kind of trade-offs around how resources are scavenged. If you're interested in this topic at all, there's older work by Yutaka Iwata when he was with Roman Stocker, thinking about there's evidence that populations can diversify with regards to how they encounter large particles and how long they stay versus dispersed new particles. There's some really recent work by my colleague and friend Johannes Kijkstra, thinking about for cells that are scavenging small resources, how long should they keep swimming? And what's the optimal kind of swimming time to keep going? And it turns out that real populations can kind of divide into two strategies. They keep swimming at the expense of their own biomass, versus they stop swimming and float around for a long time but survive for longer. And so there's all sorts of different trade-offs that emerge kind of around resource scavenging in these patchy landscapes. And so I think the stuff that we're thinking about, which has to do with group formation and what are the advantages of forming a group, hopefully kind of fits into this larger context. Okay, so with that, thank you for the great questions. I'm happy to follow up about any of this. We have a preprint that we've added a bunch of stuff to it, but unfortunately can't quite post hopefully it'll be actually out and published soon, and you can see the whole story, but you can see a version of what I talked about today if you're interested in digging in more. And I want to thank, this work was really spearheaded by my postdoc, Tom Day. Yeah, happy to take questions. Thanks.
Thank you, Julia, for the nice talk. And we had some questions already, but we have time for a few more.
Yeah.
So someone would like to have a Marco, please.
Hi. It was awesome. I really liked the talk, and yeah, sorry for the
No.
interruption. But
That was, yeah.
So I was wondering when you mentioned the fact that your Vibrio cells sometimes are sticky and sometimes are not, right? And you also mentioned the fact that the alginate that you use make, let's say, blobs that are of the order of a fraction of cell size. So I was wondering whether, so here is the limit of my microbiology. Okay, so I don't know whether the clusters that are made of different cells, meaning that they don't come because of cell division. I imagine that there are cells that get in contact with each other and they keep stuck to each other. Is that could that be interpreted as a consequence of actually individual cells trying to get sticky to keep the blobs of PEG close to them, as opposed to actually wanting to stick to other cells?
Yeah. So that's a great question. Thank you, Marco. So we think we're starting to push the boundaries, because you can imagine kind of sweeping the initial concentration of cells and looking at this transition from clonality to more aggregative growth. And we do think that the fact that you have encounters with the sticky polysaccharide, that groups are sticky. They're sticky for alginate, and they encounter other groups. And so this like group situation, I think I think you're right that it might have more to do with encountering and holding on to the alginate than encountering that's what you were alluding to,
Yeah,
right?
exactly. So I was wondering, so if I do a thought experiment where the alginate is actually fluorescent, and then I would see the cells have a sort of like corona of alginate around them.
Yeah, so they do make fluorescent alginate, and we're heading in that direction for sure. It's a little tricky because you have to wash out, you put in some, and then you have to take out this, stuff that's not attached to actually see anything. But right, I think that there's something interesting there. But it does seem to be, in a not formal sense, it does seem to be like a kind of like a coagulation that's going on, which is really interesting.
Okay.
But thank you.
Cheers.
Yeah.
Thanks.
We have a question from Rosalind, please.
Yeah, thanks for me too for a great talk. My question was also along the lines of the kind of alginate particle size, because you mentioned at the beginning of the talk how about the Ali effect and the enzymes. And so I was wondering, like in the regime you're in, if a single cell encounters one of the alginate particles, does it actually have enough enzymes to kind of rapidly digest? Because like I could imagine if a cluster encounters, it would be able to eat quicker because it's got more enzyme kind of thing.
Mm-hmm. So it's going to depend on the individual cell's ability to make enzymes. And that's definitely between different strains, that's a variable trait. Some cells make a lot of enzyme. And in that case, they seem to be able to put enzyme out into the world, have that enzyme have activity, and survive. If they're not making enough enzyme, then I think you can get into the state that you're alluding to, where it's not either, the individual cell, if there's no way to tether the enzymes or if there's no, matrix, the enzyme's just going to go out into the bulk, and then you're never, their time scales come into play with, how quickly can things be broken down, how quickly can it be recovered. And so I think there's a lot that we don't know about how those first steps for things that can form groups, about how you actually get that first division out, and how you can create that local density, and how you don't just lose enzymes to the bulk. It's continually surprising to me that these marine strains broadcast their enzymes and don't tether them to their surface. Yeah. There's great work from Rudy Aman's group and others. There are other strategies that you can break down polysaccharides like this, but for the strains that we work with, to my infinite surprise, they just let them go out into the environment. Does that kind of answer your question? I feel like I answered around what you asked.
Yeah, it's super interesting. Thank you.
Yeah.
Thanks. We have another question from the chat, I suppose. Let me check. Yeah, okay. Shlok Vatsal is asking, following on the discussion on flux scaling with R to the 2.6 and not R to the 1, could it be that the cluster is not formally packed and could thus be permeable and has a biological active area scaling differently than a solid sphere?
Hi, Shlok. That's a really interesting thought. I will say that to Marco's question, I'm not a morning person, and I apologize. This is kind of at the edge of my my understanding. I think that it should scale to the power of 2. I don't have good intuition to give you, but I agree with Marco that the diffusive case in the example that he's thinking of should scale to the power of 1. And so there's something that I'm forgetting to tell you, and I apologize. But yeah, but the packing certainly could change kind of how resources are allocated. I hope that answers your question.
Thank you. So if you don't have any other immediate question, maybe we can actually thanks again, Julia, for the very nice talk, the audience for being here. I will